So this is the flow of genetic information, and as you know, DNA is transcribed, RNA, a precursor, is spliced, and then modified at the end, so simultaneously with the splicing, then goes out of the nucleus and in the messenger in the cytoplasm of the cell becomes translated. Now, the important thing is that there are changes here at times, mutations, that produce mistakes that carry on to the protein or to the non-production of the protein. And in that case, we may have a lack of function and disease. So the errors can be in many of the process in the flow of genetic information. I mean, from the transcription process to the translation process. We are going to focus on what happens when there are mutations that affect the process of splicing. That is the more, the one that will be central. Well, RNA is central there, and this course in RNA is a lot of splicing going on. So you have already seen this in detail, so we go ahead. You saw that there are three basic elements, very sort of simple, and as John said, in yeast, very well defined. In human, they are a bit more degenerate, so there is a lot of variation, except the GU and AG. We are talking about the spliceosome, the type 1, also the more general spliceosome. GU and AG are the only ones that are 100% conserved in most of the genes, and the branch point is fairly degenerate. There is this A only that is, but it's very difficult to identify what is around. So from a point of view of the recognition, the spliceosome manage very well, but by eye we cannot always manage. We cannot manage because in addition to these splicing signals, there is a lot of accessory factors. There is a lot of these protein RNA interactions that modulate the exon recognition. So you have sequences within the exons that are splicing enhancers. So there, a protein interacts with that sequence and helps on the recognition of the splice sites. There are also splicing silencers. So they make the recognition more difficult. And these enhancers or silencers can be found also in the introns, so away from the exon, even quite far in the introns, can favor the recognition or can suppress the recognition of a given exon. These elements are very, very important in alternative splicing, for example, when you have, as it was mentioned before, a single gene that gives rise to different proteins by exchanging exons, splicing in or out. But also they are extremely important for genetic diagnosis, because mutations in these sequences that may seem just normal nucleotide polymorphisms that exist between one individual and the other may cause disease. Obviously, the dream of any clinical geneticist will be, I take the DNA, I sequence, I put it in a computer and tells me which is the possibility that these mutations or these changes are a splicing mutation and hence the consequence of disease, the consequence of the disease. I put this at the beginning, it should go maybe at the end, but obviously there is a lot of work on bioinformatics. And if you have a given splicing, a given nucleotide variation in the genome, there is a whole array of programs and applications that will predict if that mutation will affect the splicing or not. However, as we'll see in a minute, the predictions are rather unreliable, and you never will use that as a diagnostic tool or as the unique diagnostic tool. It's only orientative. I mean, it's only something that you can use to say, well, maybe, but you never can give genetic counseling advice on that basis. Obviously, there are ways to analyze, that is by analysis of the transcripts, by in vitro tests, and so on and so forth, that should certainly confirm this. And many times, this gives you the deviant information, because indicates changes that may be important, and then at the end of the day, you find that they are not. There are many of them, many combinations of them, but I show you just one example. That is a gene that is not a gene linked to a disease. It's a gene that we have been studying for a long time. Is the fibronectin gene, an extracellular matrix protein. This is an exon that is alternative splice. It's include or exclude in different proportion in different cells. If I apply most of these, any S-Finder, Rescue S, these computer programs, and to see which of these could be, which part of the sequence could be an enhancer, it comes up this. All these sequences in blue, in red, are sites, according to the program, that will bind SF2 or SC35 or SRP40 and so on. These are SR proteins, that normally modulate splicing in a positive way, binding to exonic splicing enhancers. Now, if you look at this, well, there are enhancers all over the place. But if you go and just remove this little bit here, you get, this is a splicing analysis. Better we go step by step. We construct mini genes. So a gene that contains the previous exon, the alternative splice exon, and the exon after that. You transfect this into cells, and then the RNA, the cell RNA is extracted from the cells, and an RT-PCR, so reverse transcriptase and amplification reaction is carried out, and you can see a band of a certain dimension if the exon is included, or a shorter one if the exon is excluded. This pattern here is the normal pattern, so two forms, one included and one excluded. If I delete that bit here, I get only the excluded form. Now, that means that that bit, all this is irrelevant. And the only relevant region is this one, these 8 nucleotides here. And all the bindings that you can see of these SR proteins happens there, because this is the normal sequence, and this is the deleted sequence. And none of the SR proteins are binding in the deleted sequence. So all of them bind in this region. Maybe one protein, one RNA, and you have a population of RNA protein interactions, or maybe in a synergic way, one protein binds first and the other interact with the protein. But this tells us that probably it's more a mixed population of different RNA protein complexes that modulate the positive splicing. So useful predictions, but careful with them. They are only predictions, as the word indicates. So the bioinformatic tools are useful, can also be better. But apparently what is missing is a better knowledge of splicing, above all. And to do that, we need also better tools to study splicing. I make you an example, another example about computers and experiments. This is a gene, it's called the Neurofibromatosis Type 1 gene. Mutations in the gene cause a disease called neurofibromatosis. We'll see a bit more later. If you take this region between exon 34 and exon 38, you have this sequence. If in this sequence you make a prediction of splice sites, 5' or 3', red and blue, AGs and GTs, the computer will tell you that all of this could be splice sites. Now, we know the cDNA of this gene, and we know that actually only the ones that are at the end of these purple sequences, that are the exons, are real. All the others are wrong. Furthermore, this one here, in green, if you see it, this one here, was not seen by the program. And actually is recognized 100% by the spliceosome. So the spliceosome is not only a work of art, up to now is smarter than the computers, okay? Or that our knowledge really, not that the computer. So the conclusion here is that exon inclusion depends on many factors. The strength of the splice site, so the relation of the sequence of the splice site with the consensus, is one very strong factor. And the other factors is the interactions of protein RNA at the level of enhancers and silencers. Positive and negative interactions. If you have a strong splice site and many positive interactions, you get inclusion of the exon. If you have a weak splice site and a lot of negative interactions, you get a skipping. This is useful for the alternative splicing. In some cases, you want the exon included. In some other cell types, you don't. For example, immunoglobulins have variations that are membrane-bound or not. And some of those are generated by this type of approach. In some tissues or in some cells, the same exon can be is included in some cells because that cell has a combination of positive factors. In another tissue, that exon may not be included, is skipped, because that tissue has a predominance of negative factors. It's clear? Interrupt if you want to ask in the middle, okay? Unfortunately, it's not only that. Actually, the global context of the genome, where this gene is located, the speed of the RNA polymerase when it's transcribing, and another series of factors that we call context factors may alter the splicing, okay? So splicing is determined by a great variety of things that decide which sequence is an exon and which is not. When it's decided, the spliceosome takes over and proceeds to do the removal of the introns. So let's go to some of the diseases. Splicing mutations are actually a variable component of mutations in genetic disease. Some of them are very high. I mean, about a third of the mutation described for some disease, like frontotemporal dementia, I don't know, neurofibromatosis, for example, or ataxia, are very high, between 1/3 and 1/4. Others instead are very low. But what is difficult of this is the diagnosis of this. No? If you are in a clinical diagnosis lab, normally you may have the suspicion, at times, often the certainty, by the phenotype of the patient, that that patient has a certain disease. Now, if you want to know which mutation, so you can study in the family who is predisposed to disease, who will have it, if there is a chance that the new pregnancy will be, what is the chance of the, in a new pregnancy to get a diseased child. You need to know which is the mutation in the gene. And sometimes it's very easy. I mean, in the thalassemias, for example, well, the genes of alpha globin in this case are deleted. You have only beta, and then you don't have a good hemoglobin, and that is actually hydrops fetalis is a lethal disease at birth. I mean, it dies in utero at birth. But this is, you see that there are not alpha globin genes. When you have a mutation in a splicing, putative splicing regulator or modulator, you may have a mutation that may alter the equilibrium that we saw before, weakening some interactions, increasing others, or positive or negative. So it's very difficult to know if the mutation will, unless it's in the canonical GUAG sequences, if the mutation will take you to disease. There are many defects that you can expect. I mean, if you have a given exon and there is a mutation in the G or in the AG at the 3' splice site, or in the GU at the 5' splice site, you may have exon skipping. But if there is a variation here that generates a new 5' splice site, you may have a cryptic 5' splice site that is used, or a cryptic 3' splice site. And that may put the protein out of phase, may add a few amino acids that affect the folding of the protein, and so on and so forth. You can have in the very middle of the intron a mutation that changes a sequence to a good 5' splice site, and then you get a pseudo exon inclusion. An exon that wasn't there before, was part of the intron, now is there. So all these are the type of defects that you can expect, and that's why diagnosing them from the sequence of DNA is a bit difficult. So we'll go a bit, I am going a bit fast to save time, but please interrupt if you... we'll go to some examples. I mean, how can you diagnose a splicing mutation? I mean, the easier way or the more safe way is to look at the RNA of the patient. But that is not always possible because either it's not available, you can get DNA, it's easy in any condition, get RNA from blood cells, you need to work faster, go from the moment that you draw the blood to the moment that you process the RNA rather fast and process the rest later. Some tissues, I mean, if you have a mutation that affects the brain, it's difficult to take a sample if that gene is not expressed in blood cells or skin cells. So not always is possible, but this should be the preferred one. You take the RNA of the patient, you amplify it with reverse transcriptase, first the synthesis of cDNA, then amplify the cDNA, and you get a fragment that will tell you if all the RNA that should be there is there or not. If not, you can do what I showed you before. You prepare a mini gene where you insert the mutated sequence and see if in an artificial cellular system you get inclusion or exclusion of that exon. I give you an example of direct detection of mutants. This is a study that was recently carried out in the UK to see the feasibility of looking at RNA. If any of you was involved sometime on DNA diagnostic lab or something like that, you know that obviously it's preferred to do the DNA. You sequence the DNA and that's it. Yeah. In this case, RNA was also prepared in each patient that was suspected of having the Marfan syndrome. That is a syndrome that carries mutations in a gene called fibrillin. if you sequence the RNA, you find, if you do this amplification, so across the different areas of the gene, you may get some type of signal that you don't see here. But anyway, here there are two bands while you are expecting only one in this fragment of the gene between exon 60 and 62. This means that there is one exon that probably is a skip. If you sequence this band, you know that that mutation is making that exon skip. In this case, this one. If you cannot do that, then you resort to cloning your bit and trying introducing into cells, and 24 hours later analyzing the RNA that the mini gene produces. If you use primers in the heterologous context of this gene, you may be able to avoid seeing the endogenous gene of the cell that you are introducing it there in case that gene is expressed. I'll show you this for several cases of neurofibromatosis type 1. That is a disease that has a different penetrance. I mean, you can have just an ugly aspect with a lot of neurofibroma, so you can have very vicious tumors of the nerve sheath. For example, one always thinks that an exon is for protein coding. But actually, one has to think that the exon is also coding for his inclusion. Okay? The exon should be in the messenger to code for something. If it is not in the messenger, it doesn't code. So within the exon, there are these signals, exonic splicing enhancers and silencers. In this case, NF1 exon 37, for a long time, there was a mutation here, this TAC that codes for tyrosine, to TAG that is a stop codon. So that's okay, it's a translational mutation. The protein will be truncated. But actually, if you go and look at the RNA, both in the patient and in the mini gene, you get skipping of the exon with the stop codon. But you get the skipping of the exon also if you change the C to a T. And that triplet, TAT, is coding also for tyrosine. So even if we have a mutation that didn't change the amino acid, we will have the disease because the exon will be skipped. Because that sequence is part of an exonic splicing enhancer. Yes? No, this is a mutation. This is a mutation known to cause the disease. But this one here, this one, no, this is stop, but this one is the same sense. Same sense mutation that also skips. So really it's a splicing mutation, not a... Let's move from the exon to the 5' splice site. What you are seeing here is a sort of what we call a pull down. I mean, you take an RNA sequence, short RNA sequence, for example, across the 5' splice site. And you take nuclear extracts and incubate with it, separate the complex RNA protein with magnetic beads or whatever, and look at the proteins that are in the complex. And you can see here, just on the chromacy, that there is a big difference between different 5' splice sites. So the profile of protein is different. You have NF1 exon 3, exon 37, exon 7, APOE2 exon 1 with a big complexity. Some of them is visible the signal very clearly. These proteins 7 and 8 are the proteins of the U1 SNRP. You remember the U1 SNRP is the one that recognizes the 5' splice site. So it's obvious that it should be there. But in most of the others, you cannot see it. The sensitivity of this method is not such that I can say that it's not there, but means that the interaction is much weaker. At least that for sure. So if all these 5' splice sites have different complexes, maybe there are different behaviors with the same mutation. Okay? And I want to show you a few examples. The black dots are base pairings with the U1. Okay? So the basic recognition of the 5' splice site. These are different 5' splice sites, and you can see that some of them have a very good base pairing. This one here is a mutant of this that was found in a patient, actually in the patient of the photo. There is only this change. An a G in position +5 changes to a C and reduces the base pairing from 8 consecutive to 5. But this one here, is identical to this one and causes something different, that is intron retention. And this one here, where it is, this one, have also 5 or 6 base pairings only, have a C in that position, but is included happily. So there must be something different, particularly between this one and this one, that makes this difference. If you look at the sequence now, this one is U, G4, UAA, so on, and this one is AA, GU. I point to you the reference already. These 4 Gs here, only 2 here. Why that should be important? That sequence with many Gs tend to be a binding site of a negative protein for, mostly negative protein for splicing, called heterogeneous nuclear ribonucleoprotein H. For friends, HNRMPH, okay, for short. That sequence makes that this protein binds here and reduces the amount of sequence available, or at least competes with the U1 complementarity. So really, what I am anticipating the conclusion, really, all these ones don't exist because the protein is sitting here, so maybe only a few exist of this U1 binding, and this G is central to the ones left. We'll see it in a minute in another example. You can show that because if you disrupt this G for A, the G run is disrupted, H doesn't bind anymore, and now you have full inclusion. No? So normal gene, C mutation, skipping, with a C mutation, but with an A in that position, inclusion. So the double mutant is healthier than the single one, because disrupts the binding of H and goes to C. Doesn't exist in humans, that double mutant. That's artificial. We have made it to prove the case. This to prove that the binding of H is dependent of the run of Gs. And well, there are a series of examples that confirm this. I'm going to skip them. And what I want to show you is that there are many variations on the sequence that can be related, and not always you go for sure to one thing. In this exon 4, if you get different changes at position -3, -4, and so forth, you may get that there is no binding of H, but other HNRMPs start to bind. So you may get some splicing deficiencies because you created a new sequence that is the favorite sequence of another inhibitor. Not always as the outcome is the same as well. That complicates things. This is an example taken from another disease called long QT syndrome. It's a cardiac arrhythmia that is the cause of sudden death in young sportsmen. I tell you here because this could be silent, but when you force the heart, you can have overlapping of the electrocardiogram waves and fibrillation and death. This channel, this one here, we have studied the mutation in the +6 position. So this is the GU, okay, the 100% GU. These others are the variability in the regions around the GU. In +6, only 40% have a U. Other sequences are present. But in this case, a G, a U has been changed for a T. A T has been changed for a C or for a G. Okay? What it was? A C. No, this U for a C. GT, GA, GT, this position +6, T to C. That disrupts the base pairing, this AU base pairing with U1, and you don't find U1 interacting with this. If you construct a U1 that is where you change the A to a G, you get base pairing back and you get again return. But look, here there is no skipping. Actually, this is the normal gene inclusion. This is the mutant gene. So this 5' splice site is not recognized, but this 3' splice site is recognized. So you get an intron retention. The system treats this as an exon, big exon. If you restore complementarity, you get this intron retention back to normal processing. And in this case, it's not an inhibitor binding there, like in the exon 3 of NF1, but it depends on the exonic sequences. You see, this is the mutation that we saw in NF1 exon 3. And this is the mutation that is in this channel, in this potassium channel. If I introduce this mutation in the potassium channel, I still get intron retention. So it's not this mutation that causes skipping. It's the fact that it's near to the 4Gs, you remember. In this case, causes lack of recognition. And it's due to sequences inside the exon, because if I put just the NF1 sequences there, I still get intron retention. But instead, if I put the sequence of the potassium channel, the original mutation, into the NF1 exon 3, I get exon skipping. So it's a sequence somewhere here that is deciding that this 5' splice site may not be recognized. And it's this polypurine here. There are many other sequences contributing to this, but this is one of them. Essentially what is going on. So the spliceosome sees this 3' splice site and probably looks for a 5' here or at some point, and you can have no definition of this one, total non-definition, and then this is not recognized as an exon, and the exon is a skip here. Or is recognized as an exon, is included, or like in this case, there is an exon definition of some sort. The 3' splice site is recognized, but this intron is not recognized and is left on. I think that these are other examples of cryptic splice sites. I will skip them and lead to the next talk. We have worked a lot on cystic fibrosis, and regarding the 3' splice site, we have found an aberrant splicing on exon 9 that was caused not by mutations in exon 9, but by variability in the 3' splice site. This 3' splice site has a sequence that is repeated. TG9, T9, or TG10, T7 is variable. TG, you know, is the microsatellite that people use to map genes in the chromosome. So it's quite common. And in this case, it has landed here probably because a transposon hit sometime, because only primates have it. The mouse CFTR exon 9 is 100% included and have a normal 3' splice site. Now, we found a patient that have this configuration, TG13, T3. And these make a very poor recognition of the 3' splice site. And this poor recognition causes full-blown cystic fibrosis. Intermediate recognition, like this configuration, may cause non-classical cystic fibrosis. And other variations instead give you a normal one. The reason for all this was that there is a protein called TDP43 that it was bound to the UG sequence and interacting with other HNRMPs, A1 and A2, inhibits the 3' splice site recognition. We work a lot on this protein that is very conserved from human to Drosophila, and it's an HNRMP. And recognize GU through these two phenylalanines. And it was an obscure splicing factor until in 2006 it was found to be the protein that accumulates in the inclusion of the brains of people with amyotrophic lateral sclerosis or frontotemporal dementia. And now Emanuele will tell you that story. Okay? Thank you.